So we were learning about the orienting process. So orienting process are not governed by strict stimulus response relationships. EV Holtz demonstrated that the specific physiological condition of an organism, its drive state, exerts a precise influence. Many fishes orient simultaneously to gravity and to the light. In horizontal light from the side, the fish is turned 90 degrees Celsius in response by the dorsal light reaction, while the static receptors attempt to hold the fish in equilibrium in its normal position. The fish then comes to rest in an in between position that can be measured precisely in high packed forms. For example, angelfish. The stronger the intensity of the light, the more the fish will orient toward the light. If the weight is increased in the static organs by placing the entire experimental apparatus into a centrifuge, then the influence of the light is reduced. Up to this point, up to this point, it appears as if the fish is suspended between two arms of a scale, one representing the light component and the other the static components. But there is another factor at work. Should the hungry fish perceive a prey, the orientation to the light exerts more influence, that is, optical stimuli are now stronger. This example supports the contention that orienting reactions can be depend upon an inner readiness to act, such as hunger. So, according to their function, orienting program can be classified into three main groups, such as space orientation, which encompasses the orientation in spatially oriented reference systems. So, space orientation, which encompasses the orientation in spatially oriented reference system, where the stimulus sources provide the coordinates. Then comes the stabilization of posture and movement. Then comes the object orientation, which encompasses all orientations toward directly perceived objects which are at the same time goals. Into these three main categories, it is possible to fit all known orienting capacities, as can be seen from Sean's compilation. Orientation in fixed species reference systems. Stimulus sources provide the coordinates and are served to maintain the normal position, and they are not the goal of the orientation. Orientation with the gravity receptors. Maintenance of physical balance against the pull of gravity and compensatory eye movement. Posture reflexes. Control of position and direction of action in space of the body, directing control of free swimming fishes, octopuses, crustaceans, and so on. If body appendages via proprioceptive control and of other objects by means of optical tactile perception, recognition and distinction of horizontal and vertical structures, orientation with the eyes, orientation related to light, orientation of many water animals, not quite correctly called dorsal light reaction, horizontal orientation, compass and sun compass orientation, orientation to landmarks, tactile kinesthetic registration and repetition of its own movements orientation, electrical and magnetic orientation in case of electric fishes, orientation in currents like air, water, associated stuff, and the stabilization of posture and movement or locomotion independent of locality via mechanical rotational sense organs and semicircular canal structures. Next one is with the eyes, optomotoric or the kinetic control. Then comes the subject orientation. Stimulus resources are the objects of orientation. Frequently, locating processes, goal and direction are considered. Then the optical targeting orientation, mantis or chameleon, etc. are examples. And the acoustical location, echolocation of bats, porpoises, etc. And the tactile location of dragonfly larvae, clawed frogs, chemical orientation, thermal orientation, infrared organs of pit vipers, temperature preferences. In the following sessions, I want to demonstrate the way in which orientation mechanisms work by discussing some selected examples. We shall pay special attention also to the active role of the organism in the orienting process. Comprehensive reviews on this topic have been published. So, first we are going to look at the first topic, that is the control of body position and movement in space. Control of body position and movement in space. Many fishes and water beetle larvae orient themselves with their eyes toward light, which can be easily demonstrated with an experiment. Water beetle larvae swim up to the surface to get air. If an aquarium is illuminated from below, the animal will swim to the bottom and turn their back toward it as if it were the surface. Unless the illumination is reversed, the animal will suffocate there. So, when light comes from below, Acelius larvae swim with their backs down. Horizontal swimming and attempts to get air can be seen. However, it is not as if the light stimulus is coupled, so as to speak, with the rigid obligatory reaction. The animal may orient its direction of movement toward the light, but which particular direction with respect to the light source is followed depends on its internal state. If the animal needs to get air, it will swim upward. Afterward, it will swim away from it. Depending on the internal state, different orders go to the orienting mechanism so that the animal can choose between various directions and orient itself accordingly. There is no fixed reflex like a stimulus response relationship. The organism can actively change its direction with reference to the light. This was elegantly demonstrated by H. Shawn in 1962. Disticid larvae have 6 stomata on each side from which information is integrated in such a way that the animal is able to move in a specific direction with respect to the angle of light incidence. The angles of light stimulation can be controlled by covering individual eyes and by diffusely illuminating the aquarium from all sides. The animal will then swim with its dorsal or ventral side up in circles in the case of symmetric binding, in an attempt to maintain a reference position with respect to the direction of illumination. If the angle of light incidence coincide with the angle for the reference position, the larvae stops circling. The turning tendencies, which can be computed from the circles that the animal performs, can be represented on a curve where the orientation of the body depends on the angle of light incidence produced by selective binding of stomata, so that one obtains a sinusoidal curve from which the strength of the turning tendency for a given expected position can be read. A normal larva shows a number of different behavior patterns in which it can assume various reference positions in space. After breathing at the surface, it swims downward at an angle of some degree and continues horizontally when hunting, or it waits motionless for prey. When it swims upward again at an angle toward the surface, shortly before reaching the surface, the larvae changes the direction of movement and swims backward until the tip of its abdomen breaks the surface. For each of these behavior patterns, there is a different reference value. Scientists determined the curves that indicate the turning moment in degrees of magnitude of various reference positions and found that with each change of the reference position, the curve that represents the strength of the turning tendency is shifted up or down on the ordinate. The position of the upper and lower values can be curve remain unchanged. So, characteristic positions of 8 behavior patterns in a Acelius larvae when the light comes from above are shown. Postural orientation based on Statolith organs has been studied in detail by E.V. Holtz and H. Schon. The gravity detectors of crustaceans and vertebrates consist of statocysts with a sensory epithelium. The statoliths rest on the hair of the sensory cells. The gravity detectors of crustaceans and vertebrates consist of statocysts with their sensory epithelium. The statoliths rest on the hairs of the sensory cells. Pressure downward on the statoliths and pulling in the opposite direction elicit no excitation, but a parallel shearing force does. If the hair receptors of the left statocyst in a crustacean are bent outward, a turning tendency to the right with respect to the longitudinal axis is released. If bent inward, they cause turning in the opposite direction. With unilateral removal of the entire statocyst, fish and crustaceans turn from their longitudinal axis in the direction of the injured side. E.S. Holtz thought this was due to the continuous activity of the sensory cells in the statocyst. Normally, this continuous discharges of impulses in the left and right statocysts cancel each other. After elimination of the statocyst on one side, the turning tendency in the opposite direction becomes manifest. This interpretation is supported by the results of experiments in which sharks whose statoliths were removed without damaging the sensory epithelium showed no tendency to turn. Crustaceans without statoliths, in which one of the empty statoliths had been removed, turned towards the injured side. The discharges of resting potentials of the sensory epithelium on both sides cause, according to these findings, opposing turning tendencies which cancel each other. The shearing forces in the statocysts, which emanate from the statoliths when the animal turns, increase the rate of discharge of impulses in one direction and decrease it in other according to the principle given. Many animals also orient themselves according to the direction of incident light. E.V. Holst investigated this integration of two orienting mechanisms in fish. Fish turn their dorsal side to the light and assume a slanted position toward it when it comes from the side. This slanted position is the resultant of stimulation received from the eyes and from the statolith organs. Change of the endogenous condition can lead to a changed evaluation of the optical components, which is then expressed in a different postural angle that the individual drive state enters the process that discussed.